let's jump into the magical world of gene editing through CRISPR. But what does it even stand for? Well, CRISPR stands for the clustered, regularly interspaced, short palindromic repeats. Now that we know what it stands for, let's delve deeper.
To begin with, in prokaryotes, the CRISPR system acts as an immune defense. These cells already have the gene for Cas9 in their DNA, and when viral DNA enters, it's recognized as foreign, and the cell begins building a defense against that specific virus. Now the main proteins involved are Cas1, Cas2, and Cas9. Cas1 and Cas2 work together to capture and store pieces of viral DNA, while Cas9 is responsible for cutting that DNA during future infections, and this creates a long-term defense. Now in here, Cas1 and Cas2 are bound to the viral DNA. They capture a short fragment and insert it into the CRISPR array in the prokaryotic genome. This stored sequence acts like a memory. The cell then makes guide RNA from it, which works with Cas9. Cas9 scans incoming viral DNA for a PAM site, and once it finds it, it binds and cuts the DNA, stopping the infection.
Yo, why is CRISPR such a bad barber?
Why?
Because it only cuts when it sees the payment. Get it?
LOL. Barbershop haircut costs a quarter. Okay, wait, but seriously though, what is PAM?
Okay, yeah, but in all seriousness, the PAM site is where all the action happens. It stands for the protospace adjacent motif. Well, what's the function of the PAM site? Well, it's essential for the CRISPR system to cut and cleave DNA. But where do we find it? Well, let's look at some DNA right now. You have your 3' and your 5', and you're looking for 3 nucleotides. In this case, we've given the example of N followed by 2 Gs, where N means any nucleotide. So, looking for any nucleotide followed by 2 Gs, or guanines. Okay, let's look back at our viral DNA. Here we have the strand, plus the Cas1 and the Cas2 from earlier. But, as you can see here, we've introduced a new variable, being the PAM site. In this case, they found the nucleotides A, G, and G, which, if you remember from earlier, is a PAM site. So, they proceed to cut that strand of DNA until the PAM site. So now that we have a cut piece of DNA from the viral DNA, what do we call it? Well, they're called a protospacer. Why? You'll see in a second. Let's look at the bacteria for a second. Here we have something called CRISPR RNA, which consists of spacers and repeats, and here we have our protospacer from earlier. What we see now is how this protospacer joins CRISPR RNA and becomes part of it, hence becoming a spacer and being part of the CRRNA. Now that we've discussed Cas1 and Cas2, let's talk about Cas9. So here we have our CRRNA, and we're focusing on a spacer and a repeat. This pairing here is really important because it's the structure they make within the Cas9 itself. So, as you can see, the spacer and the repeat form a bond together and make a shape much like the one you see on screen, forming what we call tracer RNA. This is really important to the structure of Cas9. Let's take a look at our tracer RNA once again. As a reminder, it consists of viral DNA from the protospacer from earlier. This taken forward, it shows us how important it is within the structure of the Cas9. Let's have a look at it. So here we split into 2 main sections. The second section here in green talks about how it's able to analyze and cut DNA strands from viral DNA. But how is this possible? Taking a closer look, we can see here that tracer RNA is embedded within the structure of Cas9. In this state, it's known as guide RNA or gRNA. This means that when referencing or cross-referencing with viral DNA, it's able to detect which ones have already infected the bacteria and subsequently cuts them before they can cause any damage. This shows off the bacterial immune system and how it's defensive against foreign DNA, which in this case would be a virus. So, to reinstate my point from earlier, when the tracer RNA is within the Cas9 enzyme, this means that it's guide RNA, so gRNA.
But you might be asking, how does the system actually work when DNA gets cut? Well, here's your answer. Once CRISPR-Cas9 makes a cut, the cell has to repair that break to survive. There are two main repair methods. The first is non-homologous end joining, or NHEJ, which quickly sticks the DNA back together, but often makes small mistakes. The second is homology-directed repair, or HDR, which uses a template to fix the DNA more accurately. Scientists take advantage of these two pathways to either disrupt genes or precisely edit them.
Let's think of NHEJ as kicking a soccer ball into your mom with your vase. You can tell it's been broken and been put back together, but it's not a really good job. Similarly, NHEJ shows where it's been broken and put back together. Let's see some non-homologous end joining in action. Here we have a split piece of DNA, and we've highlighted the PAM site here because that signals where the action is supposed to take place within the cell. Let's focus on some key players, one being Cu7080, which is attracted to the ends of the split strands. Then you have the DNA protein kinase, or DNA PK, which is going to stick to the ends of the Cu7080 and hold the DNA in place.
Now, as the proteins hold the broken ends together, DNA ligase 4 seals the gap by reconnecting the backbone. This process is quick, but it's often error-prone. It introduces small insertions or deletions. Now, these changes can really disrupt the gene, which is how CRISPR can effectively turn genes off. Now, HDR has two main repair methods: SDSA, or synthesis-dependent strand annealing, which copies DNA from a template without crossing over. And the second one is double-strand break repair, or DSBR, which uses a template and can result in crossing over. Now, before anything, let's start talking about SDSA.
Here you have two homologous pairs of DNA, one of which has been damaged. As a result, RecBCT has come to the rescue, where it recedes the DNA until it reaches a Chi site. A Chi site is a combination of nucleotides known as GCT, GGT, GG. Once RecBCT reaches the Chi site, it will stop receding the DNA, and RecA can do its job, where it will take the DNA to the homologous DNA strand on the other side, and using polymerase, we will repair it. Now you have a fixed strand of DNA, but the issue is it's on the wrong side. So what do? Well, due to complementary base pairing, the strand can return to the original DNA from which it was attained, and you now have a reference from which you can read the process or taking polymerase to fix the strand and ligase to put it all back together. Now you have a brand new piece of DNA on both sides, which is completely repaired, and unlike non-homologous end joining, is entirely correct.
Now, focusing on double-strand break repair, or DSBR. After the DNA is cut, the first step is resection. An enzyme complex called RecBCD trims back the 5' ends and creates single-stranded 3' overhangs. Now, these exposed 3' strands are then bound by RecA, which helps them invade a homologous DNA template, and in that case, it forms a D loop. Now, this allows the broken DNA to align with the correct sequence through base pairing. Next, DNA polymerase extends the invading strands, and it copies the information from the template to rebuild the missing DNA. And because both strands are extended, this creates a stable intermediate structure called a holiday junction. Now at this point, the newly synthesized DNA detaches from the template, and the cross strands are resolved, effectively breaking apart the junction. Finally, DNA ligase seals the sugar phosphate backbone, fully restoring the DNA. This process can also result in crossing over, where segments of DNA are exchanged between strands. Overall, DSBR is a highly accurate repair method, and in CRISPR, it allows precise insertion of new DNA sequences into the genome.
Alright, let's have a look at what I just said. Here we have HDR in bacterium, and what we do know here is that you need to have two strands for it to happen. The first strand appears within the actual bacteria, as you can see here, but the second copy of the DNA is made or sort of formed during the bacteria's life, thus allowing for HDR to occur.
One real application of CRISPR is treating genetic diseases such as sickle cell disease. Now there's way more, but for now let's stick to the easy ones. Now I assume we all know this, but this condition is caused by a mutation for the gene for hemoglobin, which makes red blood cells become sickle-shaped instead of round. Now, with CRISPR, scientists can edit a patient's stem cells to fix or bypass this mutation, and the corrected cells then produce healthy red blood cells, reducing symptoms and improving overall function. Now this treatment is done by using an ex vivo approach, which means that the cells are edited outside of the body. Now I'm pretty sure the next group will talk about this pretty well. So first, blood is taken from the patients, and stem cells are extracted. These cells are then sent to the lab where CRISPR editing can be performed before returning back to the patient through transfusion. Now, in the lab, CRISPR is delivered into stem cells using a method called electroporation, which uses an electric pulse to open tiny pores in the cell membrane, allowing the CAS-9 protein and the guide RNA to enter the cell. Once inside, CAS-9 travels into the nucleus and is guided to a specific DNA sequence. Then, it cuts the DNA, allowing the cell's repair system to modify the gene. Now, before the edited cells are returned, the patient undergoes chemotherapy to clear out existing bone marrow cells and make space for the new ones. Then, the edited stem cells are injected back into the bloodstream. These cells travel to the bone marrow and begin to multiply, and over time they produce red blood cells that contain functional hemoglobin, often increasing fetal hemoglobin levels. This reduces the formation of sickle-shaped cells, improving blood flow and decreasing your symptoms. As a result, patients experience fewer pain crises and a significant improvement in quality of life.